So what justifies this neuron by neuron search? I mean, I'm thinking about different kinds of network structures you might have in your brain. I mean, it might happen, maybe there are some coarse-grained networks you can already search out.
Right, so if you're a physicist, you might say, why don't we have a coarse-grained statistical description? Like, I don't need to know the position and velocity of every atom inside a gas. I've got a statistical description of it. And so I would argue, I mean, so is that the case for the genome?
Well, I don't mean it's very difficult to map the genotype level, but yeah, I mean, I agree that you need, but I just wonder.
So in the case of the genome, we can study, we could coarse grain the genome and look at the, say, fraction of A's, C's, and G's and T's in stretches, right? But we would lose a lot of information in the genome.
I agree that, I mean, the best possible data collection is if you have every single piece of possible data, but given the fact that we're still so far away from mapping out the whole connectome of the brain, I mean, are there things we can do in the interim?
Well, the interim things are to study small chunks of brain and then to map out a small fraction of the connections inside each chunk.
So have you compared, say, two small pieces of connectomes in different regions and seeing the similarities or differences?
No, what we do right now, and I could show you some of that, is most people are studying perception. And, for example, the retina is being studied by EyeWire, and we're trying to find the rules of connection between various types of cells. So it turns out that in the retina, you can take every neuron and classify it into some catalog, maybe 60, 70. That catalog is still not totally agreed upon. But we don't know the rules of connection between those cell types. It's believed those rules, the connections, respect rules between the cell types. They're not known. And it's plausible that we need to know those rules to understand how the visual computations are performed by the retina.
So, what have they found?
What's that? So there is a coarse graining in the sense that all the cells of one type are thought to perform the same visual computation. So we're collapsing the diversity. Let's say there's millions and millions of cells in the retina. We're hoping to reduce that to, say, 60 types of cells and connections. But to discover the rules, we have to look at individual cells. If you so we don't know of how to find the rules without looking at the specific cases.
So you're saying you want to study different kinds of functionality of the brain, there are different degrees of coarse graining that cater towards finding a mechanism for.
Yes, you can't coarse grain it in a naive way because two neurons that are right next to each other might be performing completely different visual computations. If you coarse grain it in this other way, where I say, I'll only take the neurons of the same type, which are spatially scattered, then that's okay.
How many hours of work have you gotten these people to put into the coloring in game?
On EyeWire, I think so far we're talking about 5,000 hours. 5,000 hours. So that's about between 1,000 and 2,000 hours per month, which is not bad actually. So we have our German collaborators employ an army of 200 undergraduates to do it, and generate about 2,000 hours a month, and pay a lot of money for that. It's expensive, and so the question is whether we can scale up by many orders of magnitude.
So how much would it? So if you just hired some people on, so the question is the scaling up. But then, how much? So what's the relative ease with which you could scale up the number of users to get more hours versus scaling up the improvement of the AI algorithm there? So that
well, I would say those go together. We want to do both at the same time for several reasons, right? So one of them is that the human input is used to train the AI through machine learning. So Daniel Berger spent 250 hours doing that, and then we used his coloring as a training set for machine learning. And in many cases these days, whoever has the most training data has the best AI. That's why Google, for example, which harvests huge amounts of human input, has very well developed AI. Not because they have better algorithms. They're using the same algorithms as other people.
Like, why should we be using this as captchas to get to know them?
Captures would be a great idea. We have to figure out, so the thing about captures is that you've been trained already to read things for whatever, all your life. Whereas here, you haven't been trained to do neuroanatomy. So we'd have to have a clever idea how to get around that. Yeah.
So do you have any ideas?
So what's your name again? Your name was?
My name is Owen.
Owen, and you're a mathematician?
That's right, yeah.
Okay. All right.
Hold on. I don't know how you do that. I was going to say, do you have any idea about how to test this slogan, I am my connector? I mean,
Well, attempting to read memories.
Okay.
So in my book, I propose I propose one experiment, which is to read the memory of a bird's song, because I've got a friend who studies the sequential activation of neurons in an area called HVC while the bird sings. So while the bird sings this stereotyped song, the neurons are firing in the same sequence every time. So the question is whether that sequence is encoded in some chain of connections. If it is, we'd be able to read out that replayed activity pattern. So, yeah, that's my proposal.
Good answer.
I've got lots of questions, but
I have a quick follow-up on that.
okay, follow-up.
Just on that kind of issue and with the general thing. I mean, if you were reading off the song, would it be like we've studied lots of birds and having looked at all of them, we take one bird from this sample and now we can say from its connectome what the song is, like if this worked. So presumably it's that rather than like, you know.
Well, it's even less ambitious than that. So what we know is that in every bird, every zebrafinch, adult zebrafinch, that in this area HVC, there's a replayed activity pattern during song. So what we would hope to do is predict the replayed activity pattern from the connections.
Yeah.
But that's not the same thing as predicting the actual sound of the song.
Yeah.
So we would have an ungrounded, what I would call an ungrounded memory.
Yeah.
So to deal with sort of if you want to ground them, you can imagine grounding the memory if you also knew the connections from that area to the vocal cords.
Yeah.
Possibly,
like
but for that you'd have to have the whole brain. We're only doing a little piece, so we'll have an ungrounded memory in the best case.
Yeah, and you have to calibrate. I mean, you have to calibrate that if you want to like read off and get a new song, or even a new motor pattern.
Well, this is the boring bird because it has only one song.
Right.
So that's why we picked it.
But presumably they vary.
But there are other.
Among the...
Well, there are other birds that'll have two songs, and there's mockingbirds which learn new songs all the time. So that's sort of increasing complexity.
So on the kind of class of questions about the game type thing, I mean, to some extent, what you're trying to do there is it would require millions, in fact billions of hours of labor. I had a quick play around with it. It was like if everyone in the world did it for a year, I think you need 3 years or something in your numbers you had there, but something in that ballpark.
For the whole brain?
For the whole brain, the whole human brain, unless I got off by a factor of 1,000 somewhere, which is quite possible. But so what you, in some sense, are saying is you need a lot of labour, and one way to get a lot of intellectual labour is the kind of game-type approach.
Yeah, make it fun.
Pay people a large amount of money is the standard approach. But you're not on your own in that area, and there's some similarity between this kind of games movement to get kind of cheap labour, and the kind of setting it home type thing for get cheap idle computational cycles. They're both ideas of there's some kind of idle thing going on. Like when people are playing a game, there's some aspect that where whether it's fun or not doesn't correlate with whether or not your capacity for thought is being used for some other project, and so therefore there's like a gap, and you can fill in that
people
gap
do
and get free stuff, right? In the same way as getting free cycles from people's computers, roughly free.
It's not only that; it's people have a hunger for Sudoku and crossword puzzles, and so if that could be used,
well, if you could make a better game than anyone else's game, that's simultaneously. That seems though that you'd have to be a good game designer as well, unless it just fell into your lap. Because people could look at all kinds of tasks they want people to do and just make them into games. But there's obviously an industry bigger than Hollywood already working on computer games and trying to get people to do them. So it could be that industry's going wrong systematically, but it's tricky. So it seems that the large thing there in both cases is that there's a kind of big economic thing where there's just a whole lot of money to be made, and it's not just researchers and kind of people with fairly altruistic kind of interests who could be doing this. Anyone could want to get a whole lot of cheap labour in various ways. You might think, I've got a whole lot of accounting that needs to get done, because we're a very large accounting firm that does a lot of people outsource to us. And we can get our accounting done for free. We've got it in a common format. We're going to turn that format and gamify it, put it out on the internet. So if this kind of idea works and can literally get you like millions of hours, if you can scale it up, then presumably other people will be able to scale it up as well.
That's
there
right.
will be a bit of a war there. It
That's
could
right.
be that it turns out that the amounts of money you could actually get out of these people is quite, or give to the people to pay them is quite low, and therefore, I don't know, whether or not it's an altruistic idea makes all the difference. And it turns out that the most cool-sounding altruistic idea is the one that's going to win this labour pool. But I'm just pointing out that it is a general question with this kind of game labour pool, that it's not obviously, it's mainly researchers who are trying to use it at the moment, but if they succeed, there may well be a land rush.
I think mechanical Turk is actually, so you have companies paying people.
So mechanical Turk's related, but it's
incredibly small.
But my plan...
It's clearly labour. Oh, sorry, it's not clearly labour. So there are people who do mechanical Turk for fun. There's
many people do it without it making sense.
So there's, well, no, not only that, but they do it as a game. So so there is like analysis on this as to there's a lot of kind of like stay-at-home mums and things in America who are doing a lot of mechanical Turk.
Yeah, but my plan is not to get...
Your plan is not?
My plan is not, no, my plan is not to do an entire human brain right now. So my plan is to get enough people playing the game that we can make the AI better, that we can do, we can build a community of game players, game designers, people that work on AI, all these people working together.
And you can obviously...
So I think there's some kind of critical mass that we have to get to.
You can obviously do some form of iteration there, but where then the AI guide they get becomes more advanced in the future versions because of this stuff, and then that might mean that those players can then get a bigger training set faster, even without increasing the population of players. So it seems to be what you're sketching. But I'm just pointing out that if you try to get bigger than that, there's some reasons to believe that it won't be a free lunch, because to the extent to which it is a free lunch, there'll be a lot of other people kind of trying to mine the same vein on that.
It's possible.
But it's interesting to kind of see it in the class of things with setting at home and making mechanical Turk in this kind of low-hanging fruit.
Mm-hmm.
How does the AI performance scale with the training set that you have there?
Well, it's not going to be just the training set. I think there's going to have to also be innovations in ways to use the training set. So we'll need brain power of computer scientists too, which we could also gamify to some extent.
Yeah, that relates to another very important aspect. Once you have this nice data, right now, of course, we're kind of eye candy. Neuroscientists will obviously be interested, but in order to actually do good neuroscience, you need to kind of figure out statistical relationship. Actually try to figure out, okay, this is schizophrenic connectoma, this is a non-schizophrenic, where are the differences? You need a lot of tools to investigate that, and they don't exist at present. So we need to
So here's the basic premise is very simple. If we're generating a petabyte of images every 2 weeks, there's just not going to be a substitute for having a lot of human brain power working on it. In the near future, there's just no way. Even if the AI did all this stuff that we just saw, this coloring independent, really rapidly, there would still be a lot of interesting observational work to be done by people, I think, because we don't know what we're looking for, and so I totally believe in this idea of mobilizing people beyond the traditional scientific enterprise to handle the ridiculous amounts of data that are going to come in. So AI is just one part of the puzzle. Gamification is just one part of the puzzle, just one incentive.
Can I ask about the accuracy of this? It's a really interesting process. You talked about you get a lot of people to look at one little part of the connectome, and then hopefully enough of them agree, and you take that agreement.
Yeah.
And I imagine you're also got somebody going through and
Yeah, sure. So that's the social computing research, which is how to get accuracy, wisdom of crowds, that kind of stuff.
It seems like
let's move beyond the so we don't have much time left. So let's move beyond the mechanics. Let's think about so we've sort of talked a lot about execution, and I'm just curious about what you comments and questions you have about more expansive issues. Since we have some philosophers here, and in general, this is supposed to be an unusual forum, right? So yeah.
Mine's I guess a more mathematical question, but I mean there are mathematical tools for trying to pick out relevant bits of structure of large data sets and things. Have you used these, or I mean I guess you don't yet quite have the large data sets. But.
Yeah, if we had graphs, then we would have if we really had turned the images into graphs, then we would have all kinds of tools from.
Okay, do you know how to turn the images into graphs after that?
That's our problem. That's exactly our problem. We have to trace all those wires and find this.
But after people have coloured them.
Yeah.
Now at that point, do you know how?
Well, then we have to find the synapses too.
Okay, yeah.
And then we have a graph.
Do you have a synapse detector?
No, we haven't worked on. We should work on that. So right now we haven't worked on it because the tracing of the wires is more time consuming. But as that gets faster, then we have to start working on the synapse detector.
I'm myself very curious,
Yeah.
as you might have noticed in that white paper on brain emulation, about how much could you infer just from these electron microscope photos about the nature of the synapses. Could we tell whether they're excitatory or inhibitory, for example? That's something I'm really curious about.
Yeah, to some extent we can, and we can also tell that from the neuronal morphology. Yeah.
On the question of whether your connectome in some sense captures enough information about who you are, I mean, I'm somewhat convinced by the stuff about people's electrical activity stopping after falling through the ice and kind of being restored.
Yeah.
To think that the electrical activity dynamics is not required.
Yes.
But I'm wondering, how do these networks of the connectome form? Is it that a whole lot of things just form randomly everywhere and then they atrophy in certain bits, or?
Yeah, we don't know, and that's
If not,
yeah.
if they just basically form randomly and there's not a process of atrophy or something like that physically changes based on the ones, then it's just going to be random stuff.
So if you read my book, if you read my book, you'll be shocked at the level of our ignorance. So if there are some, it can be really. So we know that long-range connections are not random. So if there's a genetic program which tells this area of the brain to send its axons to particular other areas of the brain, but not to all of them, right? But once those axons, once we're talking about where the axons are, do those axons make connections at random on any neuron inside that area? Or is there some order to it? And there are people who take one extreme who say that it's totally random. And there's other people who say that it's extremely ordered and we just haven't been able to see that yet.
I mean,
and there's theories. So there's neural Darwinism. Neural Darwinism says that synapse creation is random, but elimination is not.
Yeah, that's what I meant by the atrophy type.
Yeah, so that's why I mentioned it. Yeah, you said atrophy, right.
Okay, and that's at the synapse level, not at the neuron level.
Well, no, because it's plausible that elimination of a lot of synapses could be the trigger for collapse of a branch.
Yeah, okay.
No, these are all speculations,
yeah.
But
obviously they're very closely related to the question about do you need more data? Like, do you need to see what's going on inside the channels, inside the branches? Like, do you need to know stuff about?
Well, we do need to know that. We do need to know. So in the book, in the last chapter of the book, I discuss this. I revise I am my connectome, and then I propose a revision which would be I am my connectome plus models, biophysical models of each of my cell types.
Yeah. Okay. So, but just to like 100 or so cell types, or
in a person,
yeah.
There's already over 60 in the retina.
Yep. So what's your kind of, what would you think? Are we thinking hundreds, thousands? Are we thinking millions?
I'm thinking.
One of those is very bad news because the amount of science you have to do is like really bad.
I'm currently thinking in the cerebral cortex estimates are thousands.
Okay.
But who knows? Who knows? And the whole notion of cell type is potentially. Some people don't even believe in cell types. They say, the cell type thing is overblown. It's really a continuum.
Okay.
And yes, cells differ, but they're not really discrete.
Well, at least then there might only be several dimensions on the continuum, and that might be all understandable again.
Yeah. So the question in the end is sort of if I have a biophysical, if we think about the biophysics of each cell, is there some low-dimensional way, some low information complexity way of describing that? And we'll find out, for example, in the retina. That's the first place maybe where we'll find that out.
So, yeah, I mean, sadly, it could be difficult to work out. I was thinking you might be able to work out from this data whether it's random or not, except the problem is that if it's like highly compressed it's going to look random. So, as in, if you know what I mean, it could be difficult to tell.
Well, we already know there's deviations from the random hypothesis. We just don't know what the overall balance is yet. But that's one of the reasons we need connectomes, just to answer this kind of question. And I think there's going to be a lot more order than people thought.
So how far are you from having the retina mapped out in this way?
As you can see, not very far, right? We only have a few cells mapped. I think the paper that was published on this data set had 30 cells, a paper in Nature, 30 cells out of thousands in that retinal sample.
And so the retina is like a big disc or something, right? So presumably
It's a sheet. Yeah, it's a sheet.
So if you just want to sort of slice through it, like a cylinder or something, to understand what the retina does, you might not use
Yeah, so the image that we're looking at is a square of the retina that's 300 microns on a side. And so what we want to have inside that square is multiple examples of each cell type. So there's some cell types that are very dense. We have lots of examples of them. There's some cell types that are very sparse. Then we don't have many. So we're 300 microns is good enough for many cell types, but not all of them.
Okay. So what kind of time scale would you conjecture that you would have?
An entire retina?
Well, enough of the retina that you can you don't need more in order to be able to figure out.
Well, it depends on how fast AI progresses in the next few years and how good we are at recruiting, scaling up the game to get people to help. Right? So those are uncertain questions, right? I'm optimistic, but you could differ. Let's say the AI increases by a factor of 2 every year, and let's say, actually in short-term gains, you could imagine that our human effort could scale up much more rapidly than the AI. Let's say we made a game, and let's say that it was really successful. The number of users might go up by a factor of 10 in 1 year. I'm not sure the AI will increase by a factor of 10. That's pretty aggressive, right? To reduce the error rate by a factor of 10 in 1 year.
In terms of the labour thing, you can just work out by pricing the labour, like how much it would cost to buy this labour if it wasn't fun. Which, if I was right, it's around about 1 million person years, then most people would work for something like $500 or $1,000 per year because they're very well hundreds. And so, it's something like $1 trillion to just brute force the labour. Not that you could do it very quickly, because it would take at least a year, having everyone working on it, nothing else.
So Nick brought up the issue of...
It's bad news, but it also shows that with the game thing, that it can never scale the whole way up unless everyone's playing this game 24/7.
Well, there's feedback. You get...
Yeah.
So Nick brought up the issue of risks. What bad things could happen from... So Nick said, why do you want to speed up the process of finding connectomes? Maybe it's going to be bad for us.
Have you read Robin Hanson's What If Uploads come first?
I think I've read a few pages of it, and I've talked to him about it, right?
That's one of the potential bad things.
Right. So the risks or implications of whole brain emulation or uploading, it sounds like it's been explored. But what if, I think that's many steps beyond the connectome. So what if just the connectome itself? I think that hasn't been really considered. And in fact, my father said that was a weak point in my book. I kind of just brushed it off.
Or reading people's memories.
Well, hang on. You only read their memories if you can cut up their brain and kill them. So that's the bigger issue.
Although there's a danger of that.
You can find out what these famous embalmed, what Lenin's actual beliefs,
But obviously you don't get that because that's the connectome plus memory decoding, which is an additional thing.
But I think reading ungrounded memories will happen much more rapidly than reading grounded memories. So
if you succeeded more than expected, I guess it would maybe add some support for the chronics movement or the plasticization. But whether it would be enough to actually make it take off, I'm not sure whether it would.
Or the opposite. I was thinking if they have something negative, it could, because a lot of the ground movement is based on a lot of assumptions.
Yeah, now I'm just thinking that if it's negative, it means you couldn't, by just studying what remains of the connectome from the point of view of what we see in an electron microscope, put it together. It doesn't mean that the information is not still there. It might just be you need to dig down to the molecular level and require superintelligence to sort of sort it through and put together.
That's right. So some people say you need to have more than the connectome. You have to have the identities of the molecules, the locations of all the molecules inside the neuron.
No, but I mean, like, if the freezing process sort of scrambles the connectome, so it's all...
Yeah.
It might be like a broken up...
If it breaks the wires, or then...
It's like it might be a vase broken into 1,000 pieces,
Sure.
but with sufficient patience, all the information is still there. You just need to sort of apply more computing power and intelligence to putting the synapses together again.
That sounds like it would be really easy to check. Sort of just plastinate a mouse brain and slice it up and see if you can reconstruct.
In a sense, of course, the implanter is already a plastinated mouse brain in a sense.
Well, not whole brains.
No, that's true.
Why didn't you start with the really little things that you showed us that they'd scanned in the 70s if you want to do that?
You just need to take a tiny bit of, say, a frozen mice brain and see if it's degraded.
Well, that's what Ken Hayworth is doing with the Brain Preservation Prize. You guys have heard of the Brain Preservation Prize? Just for...
Do you have any chance of finding the dynamics of the connectome? Because this is all very static.
That's right. So we're looking at a snapshot. And we don't know how to find the connectome. A movie of the connectome is impossible right now.
Is there something I don't understand about the term, the connectome? It just seems, I guess maybe it's a little but it just seems very much like you're talking about a connectome.
Right, a connectome.
They're all totally different, or at least with the fine structure.
Yes, you're right. No, it's really connectomes.
Question about what's supposedly you have my one and Nick's one, a whole lot of people's connectomes, and then we kind of want to work out how much they've got in common, say to do relative compression on them or something like that. Then there'd be some interesting question about how much there is in common at what levels of structure and stuff. And that would seem to be something which is, in some sense, of more scientific interest, in the sense that science studies generalities rather than studying a huge amount of active postron. We want to know about what is it that connectomes have in common.
Well, in the retina, we don't actually even from one retina, we have many instances, right? Because we have many examples of one cell type. So it's a repeated structure. So, but you're right. In other things, we may need we will definitely need multiple connectomes.
And even then, it's very hard. It's not clear how you could ever do it. Like once you've got these really crazy.
Okay, so just for fun, I'll show you.
Once you've got the graphs, it's not like that.
That's a number of the program.
Yeah.
So this is the data set from iWire, the entire data set. I'm showing it to you in our desktop program. So it's 3 orthogonal views, X, Y, Z, Y, and X, Z. The retina is like, you can think of it as a club sandwich. So there's 3 layers of cell bodies. The photoreceptors, let's call it 3 layers of cell bodies. One layer has the photoreceptors, and the other layer has the ganglion cells, which are the output cells. And so the cell bodies are the bread, and then the meat between the slices, that's where the branches of the neurons are. They're all wires and synapses. There's the outer plexiform layer and the inner plexiform layer. And what I'm showing you here is the inner plexiform layer. And you may be able to see there's cell bodies here and cell bodies here. Can we turn the lights down, actually?
I think we're over here somewhere.
We can switch with the...
That's fine. Some lights also. Do you want to try first try this?
Go for it. Yes, yeah.
Okay. So you can see here that do you see the cell bodies? Like right here at the cell body. And we can move through slice by slice, but you can't really see the branches very well. So I'll zoom in more until you see the branches. All right, so that's to find the retinal connectome, we'd have to trace out all the entangled spaghetti strands. And this shows just a small color right now are the branches of some neurons by some combination of human and machine intelligence. So here's one. See this neuron right here? You see how flat the neurons are? So that inner plexiform layer, that slab of wires, is divided into many sublayers. Nobody knows exactly how many, and so each of the neurons actually produces dendrites in some set of sublayers. And so you can see that all those branches are inside here. We had to trace them all.
There's a lot of complexity. Does it do anything other than contrast enhancement, the retina?
Yeah, so the classic story of the retina is that it's a contrast enhancement device. It does a center surround linear filtering. That's what ganglion cells do. But the fact that there are, say, 15 different kinds of ganglion cells suggests that that's naive. If all it did was contrast enhancement, why would we need 15 different kinds of ganglion cells? So, for example, this one is direction selective. So this cell right here, you can see it has a very distinctive shape. So it was a surprise to find because all of its dendrites are on one side of the cell body instead of symmetrically arrayed. And the dendrites of every cell of this type are directed downwards in the retina. So there's a nice picture of that. Well, I could show you a picture of that, but there's a nice picture of that because someone has genetically engineered a mouse so that cells of this type are fluorescent. And you can see that they're all pointing downwards, which makes you believe in the idea of cell type. And they all are activated by motion in the upward direction. And it's an unsolved question as to whether the directionality of these dendrites has anything to do with the visual function that I just described.
So do we know from a functional point of view whether the retina does anything else than contrast enhancement? I mean, we can see with all this raw material there for computation, one might think it should be able to achieve something more. But have we actually...
You're right. So the interesting question here would be to use the genetic control to disable all the neurons of this type and see whether it has any impact on the mouse's behavior. Right?
Is there a medical condition they call blind sight, where you don't see, you test?
Yeah, blind sight comes from damage to the primary visual cortex.
Okay.
So you are blind, except there are pathways that go to subcortical areas, which enable you to see without being conscious of it. Seems like the information has to go to the cortex for you to be for conscious vision, but there's still subconscious vision going on. And so a lot of these types of cells may be going to those old visual systems. So here's this ganglion cell, and I'll show you. I mean, once you start looking at the richness of this, it's hard to believe the simplistic accounts of the retina.
When you say it grows down, do you mean as in like down in the visual field? Like, so it grows like away from the center of the earth, like.
Yeah.
Okay.
Down on the retina. Upward in vision. Yeah, so it's sensitive to upward motion because it's inverted vision.
Yeah.
So the red cell is a starburst amacrine cell.
Do we just guess that it's sensitive to upward motion, or have we experimentally determined that? Because I can see why it would be, because there's a time delay as it goes across it, so it correlates.
No, no one guessed it.
Okay.
People put electrodes into that cell and applied visual stimuli and found that out empirically. This is another kind of retinal neuron, this red one called a starburst amacrine cell. As you can see, that it has a much narrower stratification. So it's more flat than this other cell. And one of the interesting debates is how much this cell, is the starburst amacrine cell actually making synapses onto this JRGC, which is the purple cell. And that's critical because the dendrites of the starburst amacrine cell are direction selective. So it might be that, if they're connected, it might be that this purple cell is direction selective just because it receives input from another direction selective source. So that's the kind of question you can answer. And I don't know, let's see. So that's the reality of neural networks. It's just kind of shocking what it really looks like.
Does it look like it's outside of the retina? Or,
What do you mean outside of the retina?
I mean, other bits of the brain.
It's even more three-dimensional out there.
Yeah.
There is
The retina is highly ordered
Yep.
because you can see how this planar cells and then restrictions to sublamina and so on, and the stereotypy that we can divide cells into morphological cell types. And
it's also just a subset of the cells, like you don't have the filling in.
Yeah.
oh yeah, look how if we had everything, this would be totally packed, like the cube that I showed you before. This is just tiny. This is tens of cells, and the cells are different types. Like look how small this one is. There's a small one right there. That's a bipolar cell. And, it is, there's, man, I should, let me see if I can show you. You guys want to see the real science. You don't want the,
So the retina is, Moravec was arguing that the retina was a good model because it was highly optimized, that we understood it.
So that's the old retina is 5 classes of cells: photoreceptors, horizontal cells, bipolar cells, amacrine cells, ganglion cells, simple contrast enhancement. And the new retina looks like this. So every one of those 5 classes has been subdivided. Here's a dozen different types of ganglion cells, a bunch of amacrine cells, and it's hypothesized that each of these has a different function that's pretty nonlinear, maybe highly nonlinear. And here's that JRGC, the cell you just saw. You can see here's a genetically engineered mouse. Beautiful picture, right? All those neurons are sending axons to the blind spot where the optic nerve comes out, and they all have their dendrites directed downwards. So using that, the physiologists can stick their electrodes inside those cells and measure their responses to vision and find that they have similar response properties. So it really, the ideal definition of cell type combines all 3 perspectives in neuroscience. We have physiology, which tells us what the visual function is. And then we have anatomy, which tells us the characteristic shape and connections of those neurons. And then we have genetics, study of molecules. So all these cells are labeled here because the fluorescent protein its expression is under control of the promoter for a cell adhesion molecule. So the fact that these cells are all cells that express this gene, and by genetic trickery, any cell that expresses this gene is made to express the fluorescent protein. So that's how you get this.
It's neatly specific if we're all pointing downward for JAM-B. So one would imagine that it would be a jam C or D cortex.
Oh, so why is it that, yes, nobody knows why would this molecule be specific to these cells? This is a cell adhesion molecule, which may be important in establishing connections. So maybe this molecule is specific to generating the proper connections for this type of cell. So there's a lot of rules and order here that we don't know yet, but we're going to map out. And so the retina is really interesting because that's where the neuroscientists always talk about reverse engineering the brain, right? This is a system where we're truly going to be reverse engineering it because we can find the whole parts list, we'll have genetic control over it, we'll find the connections, and we'll measure the visual function of every single type.
One question that we've been wondering about in whole brain emulation was whether from these scans whether you could work out from slices and so on what all the different cell types are, or whether, say, perhaps for each different one there's a stain which would let you do it, but there's no way to simultaneously stain everything such that you can work it out or something like that. I was kind of interested by what you just said then, because it suggests that if you keep these slices, on hand somewhere, that maybe by doing some kind of chemical assays,
people are talking about that. Yeah.
You can find some JAMB or whatever.
Well, the dream.
And then you can say, well, JAMB molecule only occurs in that type, so we know where it is. Now let's colour that one red on our diagram, colour all the similar ones red on the diagram, whatever.
Yeah. So ideally, what we would have an image of the retina, in which we have the shape of every neuron. We have recorded the visual responses of every single neuron. And we know the entire transcriptome of every neuron. So we know which genes are being expressed. And
but some of that you only need to do once, right? Because for every genome type, you have to have done it. And now it's done for that one. We know that JAMB, because we've made all the JAMB fluoresce, we know it was nowhere else because we checked.
That's right.
And so that, once you can do one experiment like that for each type, maybe 70 types or whatever, then you're in a situation where the number of chemical assays you need to do, whatever, is not one per pixel, but one per neuron, which is a lot better. Maybe it's not enough better.
Yeah, so the ambitions of the molecular people are to produce a mouse for every one of these cell types.
Yeah. That's not so bad, right?
Yeah.
It's not like a mouse, for every neuron there is a mouse. I mean, that's bad news.
Yeah.
It's like for every neuron.
Well, as long as this list remains, doesn't expand too much,
Yeah.
but it's still expanding actually.
Yeah.
It's still expanding. So, but we have hope that it's going to be finite.
Yeah,
it's not.
exciting.
So, is there any chance of getting more information from these slices, like the chemical information?
Okay. Yeah. There's a lot of wild ideas out there. Some people want to do in situ DNA sequencing. So just right there's RNAs and DNAs all over these cells, and let's just, instead of grinding the whole thing up and sequencing those molecules, we actually sequence them in place. So we keep the spatial resolution. I don't know how long that's going to take, but that's the kind of crazy ideas people have that probably will happen at some point. So doing proteomics would be harder. I'm not sure if that would be possible. People working on that with mass spec. So you know about mass spectrometry. You can measure the masses of the things. There's something called MALDI, imaging mass spec, where you drive an ion beam into a piece of material and it crap comes off it, right? And then you put it through mass spec, and you use that to figure out what molecules are at each location when you're scanning. So there's going to be a lot of progress in imaging, definitely.
And what's the resolution on that kind of thing?
Still more optical, I think. It's closer to optical resolution.
Could you definitively work out at that resolution that it's a particular neuron? Like, so you've got your image, you know what coordinates to fire the gun at in order to get.
Well, if all you cared about was the cell body,
yeah.
If all you wanted to know was the molecules in the cell body, yes.
Yeah,
but if you want to find the molecules in the branches, then optical resolution is not enough.
If you're just typing them, then you shouldn't need that, right? Because you just find, like, it's got I mean, it's got J and B or whatever, like in the cell body for that thing.
Right.
So you know which bits of the branch are connected to which cell body because that was what you did with your
That's right.
game or whatever.
Right.
But
Right, but the thing is that the molecules expressed in the soma may not be the same as the molecules expressed in the.
But you'd at least get that.
And we know that. We know. In some certain cases, we know that. For example, in many neurons, the axons have a particular mix of ion channels for generating action potentials, but the dendrites don't.
But all you would need is for every one to find something which is unique to their cell body. And then you'd have the current resolution.
That's right.
To actually just do this.
That's right.
Yeah.
But we have to believe in the notion of cell types.
Yeah. Yeah. Yeah.
So that, if that's true, then things are a lot easier. If there really is a dictionary. But biology could be really complicated. It might be that there are some cell types, but there's also a continuum of other cells, et cetera, et cetera. We don't know what's going to happen.
Do we know, so with regard to figuring out whether a particular synapse is excitatory or inhibitory,
Yeah.
how easy would it be to check that, either just from the electron?
Well, with cell types, that's not hard. As long as you have, like in the cortex, pyramidal neurons, once you have the neuron's shape, you can say this is the pyramidal neuron.
Right.
And therefore, all of its synapses, the ones it makes on other neurons, are excitatory. And they're glutamatergic.
So for each cell type, they either have all excitatory or all inhibitory synapses, or?
Well, they're... Okay, so this is called Dale's law. So Dale's law says that a neuron secretes the same neurotransmitter at all of its synapses. So if a neuron secretes glutamate at this synapse, it secretes glutamate at this synapse.
And
Now, that's Dale's law version 1. Now, it's usually the case that if it secretes the same neurotransmitter at all synapses, then it's excitatory at all synapses or inhibitory at all synapses. There are exceptions because the receptors might be different at the two synapses. Right? So even it's possible that one neurotransmitter could be excitatory at one synapse and inhibitory at the other synapse. Because the receptors can be different.
If we know what the types are of the one it connects to, and so on, then we could work.
That's right. If you know the type, that's right. If you know the type of the recipient neuron, then you may be able to figure out the type of the receptor.
is there like a Dale's law for the receptor of the neurotransmitter?
Yeah, that's what you need.
It's, I don't know. I don't think that's adequately known, and people are working on, like people like Stephen Smith are trying to figure that out.